Thank you all for being here. Hello, Madrid. Hola, Madrid. It's a pleasure to have you there. On behalf of Rafa Bañares, who is in Madrid, and myself, and also thanks to the support of Gilead Sciences, the CIBERET, and la Asociación Española para el Estudio del Hígado, we welcome you to the Liver Seminars program, which starts today. And during 1 year, we'll have every month one keynote speaker in the field of liver diseases. And I think we can be proud of this seminar because we have completed already all the program for the whole year. You can see in this slide the speakers that have confirmed that will come to Barcelona or to Madrid to give a seminar, but also to spend the whole day in the institution. So today we are honored to have Frank Take with us. In October, we'll have Rajiv Jalan in Madrid. In November, Professor Nolt here in Barcelona, Manuel Romero in December in Madrid, Mala Maini in January here in Barcelona, Bernat Fromenti also here in Barcelona. We move to Madrid to have Maria Restigno in March, Stuart Forbes here in Barcelona in April. In May, Liver Regeneration, Jean Campbell coming from Seattle. And we will close with Pierre Bedosa here in Barcelona. So I think it's quite an attractive program that has been possible thanks to the support of these three institutions. So I think we are so pleased and we thank you, Gilead, CIBERET, and IAC. Of course, please check the website of the Liver Seminars program, www.liverseminars.eu. There you will find all the information about the speakers, how to book a seminar, a private meeting with the speaker. I think Frank can tell us now that he has been the whole day meeting different researchers from this institution. It's important to keep clear that this program is open to any researcher in Spain, not only to IDIBABS or Gregorio Marañón, anyone. Today we have people here, I can see people from Alicante, from Vall d'Hebron, from different institutions, from the University of Barcelona. So this is the spirit of this program, to have people from all the institutions. And also you can tweet Liver Seminars, you can follow us, and also in LinkedIn with Liver Seminars. So having said that, I think the important part now is to talk about science and to have to introduce Professor Frank Take. I don't think it's necessary to introduce Frank Take to this audience. He's a professor of medicine at the University of Aachen in Germany and is the vice secretary of EASL, which is our society in Europe. So thank you so much, Frank, for being here. I think we have enjoyed a lot your visit of today, and we are looking forward for your seminar. Thank you.
Thank you very much, Jordi. Thank you very much, Per, Jaime, Juan. It has been a true pleasure to come here to Barcelona. Of course, it's a huge honor to open this prestigious series of seminars, so I hope it will get even better throughout the year. But let's start with liver macrophages, or hepatic macrophages in liver diseases. And many of you do basic science projects or look at clinical samples, and one thing that you notice is if you use a model like, for instance, CCl4, carbon tetrachloride, which we inject IP, I learned that most of you use it per gavage or inhalation, that you see around the fibrotic septa a lot of inflammatory cells. And if you do staining for them, you actually realize a lot of them are macrophages, for instance, stain positive for F480 in mice. And if you have a closer look, you appreciate that not all of these cells are the same. They have different markers. Here shown as C11b and F480. That tells you that these cells are actually quite different. And I think a very traditional view on the topic would be that you have resident macrophages in the liver, the so-called Kupffer cells or traditional Kupffer cells, and you have monocytes that circulate in the bloodstream, come from the bone marrow, and can go into the liver. And altogether, those cells then form the pool of macrophages that can have very different functions. They can have pro-inflammatory or also anti-inflammatory regenerative functions. And I think a very simplistic view is to call it M1 or M2 polarization, but this is probably not the whole truth. There's part of it is probably true. Nonetheless, what became clear over the past years is that the macrophages are of different origin, and that also determines partly their function. You have cells that self-renew in the liver, and this is what I personally like to call Kupfer cells, because they are seated in the liver during embryogenesis, self-renew, and actually adapt also to the phenotype. And you have cells that are freshly recruited, inflammatory macrophages, or inflammatory monocyte-derived macrophages. Nonetheless, I think all these cells are very, very plastic, so looking at them at a snapshot will never tell you the whole truth, because they vividly adapt to the microenvironment. And this has become very, very clear. There's, for instance, one paper here in Immunity that I recommend you to read. They use the human monocyte-derived macrophages, so you isolate from peripheral blood macrophages after 3 days, and then they expose these cells to different stimulus, like interferon, gamma, IL-4, that would give you M1, M2 macrophages. Then they added free fatty acids, TNF, different stimuli, and did transcriptomic analysis, more than 300 samples. And they can actually show that there's almost never an M1 or M2 macrophage, but the macrophage adapts to the microenvironment. If you add more factors to it, they completely shift their phenotype. And this is something what very, very likely happens in vivo. And this also determines, which is then, of course, of most relevance with respect to liver diseases, the function. So the cells can actually promote disease, or they can also help to regenerate. And this, of course, makes it very attractive if you think about therapies. If you have a cell that has the potential to heal whatever injury is there, or that could maybe be inhibited, that might be a really interesting field for therapeutic interventions. So I think I would like to present you from 3 different areas of diseases, different models from our lab on acute liver injury, then on chronic liver injury, and of course this is now, NASH is the upcoming etiology, so we should probably focus on that. And of course in Barcelona, never without hepatocellular carcinoma. So let's start with acute liver injury. If you think what's the function of macrophages, I show you a very simple experiment. So these are primary mouse hepatocytes in vitro, and we exposed them to acetaminophen, paracetamol. And this is what happens. So all the cells die immediately. And this is what also happens in an acute liver injury. And the role of the macrophages is to recognize this injury to the liver. So I think primarily the Kupfer cells are the cells located in the sinusoidal space, but also with close interactions to endothelium, to stellate cells, to hepatocytes, recognize the injury, and they then aggravate or activate the immune system. And one thing that you can see in mice is if you inject acetaminophen, CCL2, a chemokine that attracts monocytes to the liver, is vividly upregulated within a very short time, and the primary producer is the Kupfer cell. So by this mechanism, the Kupfer cell attracts new monocyte-derived cells to the liver. And one thing that we have been working on is to actually try to visualize what is happening there. So we use an intravital multiphoton microscopy system in mice. And I think I show you a couple of pictures how we do it. So one important thing is for us to have a stable anesthesia. So we do it similar as some of you work also in the intensive care unit. So we do a tracheotomy, do an ECG, give an infusion with a bit of glucose to have really very stable anesthesia and have almost a normal ventilation, then a perfusion of the liver. Then we open the mouth, flip out the left liver lobe, embed it in sort of an agarose that would allow 37 degrees of body temperature. And then this whole setting is actually transferred to the intravital microscope, and we are able to actually look a bit deeper into the liver, as it can be done with disc spinning microscopy or so, where you stay really at the surface. And if you do this, one thing here in this picture, you see a healthy mouse. In red, you see Kupfer cells that have phagocytosed a latex particle, and in green, you see CCR2 expressing monocytes. And I hope you can see it a little bit. This is like 4 hours of video compressed. So you see cells are going through the liver. The Kupffer cells stay exactly where they are. So they do not move. They just stay there and sense injury if there is one. And if you look after acetaminophen, you see the Kupffer cells and the monocyte-derived macrophages are still completely separated. So you don't see any yellow cells that would have both markers. But you see that the monocyte-derived macrophages actually go into the liver and form clusters around areas of injury. And if you don't allow this monocyte recruitment to happen, for instance, if you're a CCR2 knockout mice, you actually lack specifically this one population of infiltrating cells that is here highlighted in red circles in the FACS analysis, or they're numbered. And actually, if you don't have this monocyte wave coming into the liver very early after the injury, you actually do see less injury. So at the beginning of the injury, it's really a clear aggravating function of these inflammatory cells that come in. And if we do a cell sorting, separate the different monocyte macrophage populations from the injured liver and compare it to healthy liver, or compare the different populations, you can see, although you might not be able to read all the small factors, everything that helps to recognize injury, pattern recognition receptors, chemokines, not so much the cytokines that do the effector work, but especially everything that helps to recognize injury is heavily upregulated in the monocyte-derived cells, so in the cells that are freshly recruited to the liver. And we believe that this also happens in human, not only in a mouse model. For instance, if you look at patients that require liver transplant for acute liver failure due to acetaminophen, you do see a lot of CCR2 positive cells scattered throughout the liver. And if you do co-stain, for instance, for inflammatory macrophage markers, you do see a co-localization. For instance, here S100A9, the part of the calprotectin molecule, is really co-localizing with CCR2. So in the beginning, in an acute injury phase, those cells really aggravate the liver injury and would be probably possible to impact their recruitment if you want to diminish the extent of the inflammatory reaction. But of course, the interesting part begins if you want to translate this into a chronic liver injury model. And I don't have to tell this audience that obesity is a growing problem worldwide. You see here the mean BMI has increased over the last 40 years tremendously, and the amount of people affected by obesity is really increasing. And this translates into morbidity and mortality of the liver. So about 25% of the population, I think in Spain it's a bit less, in the EU is affected by fatty liver disease, and about 10% of these patients actually have NASH, the non-alcoholic steatohepatitis, which renders them at risk to develop cirrhosis, fibrosis, or hepatocellular carcinoma. I had the pleasure to work a little bit with Homi Razavi, who is an expert for modeling how this will project. So from the obesity that you see now in children and the increase in the general population, he can project how the number of NASH and FLD patients will increase. And actually the projections for Spain, like for the next 15 years, is like this. So you have about 10 million people with non-alcoholic fatty liver at the moment in Spain, of which a relatively small fraction, about 1 million, has NASH at different stages of fibrosis. But this will tremendously increase, maybe not so much the total population, which will increase from 10 to 30 million, but the number of patients with F4 cirrhosis, NASH cirrhosis, will triple in the next 15 years. So this is something, especially for the younger of you, who will actually witness this if it all holds true and we don't take any measures. So how does it look if we look at macrophages actually in NASH cirrhosis, and if we do the staining for CCR2, which is a chemokine receptor expressed by the infiltrating monocyte-derived macrophages, you see that there is actually, especially around the periportal areas, accumulation of these CCR2 positive cells. And if we do it in a more statistical way, it's especially in fibrotic and cirrhotic NASH where we do see those monocyte-derived macrophages. And actually, if we do our co-staining here with S100A9, these cells are also the pro-inflammatory macrophages. If we look in our mouse models, for instance here, chronic liver injury model, the picture with Kupffer cells and monocyte-derived macrophages is not so clear. So a lot of the green cells actually acquire a Kupffer cell-like phenotype that makes them capable of, for instance, phagocytosing latex particles. So they look yellow in this picture. Nonetheless, if we sort again Kupffer cells and monocyte-derived macrophages from the liver in a liver injury model, we do see that actually a lot of genes, although they all express F480, they all express CD68, a lot of genes are really differentially regulated between those two cell types. And of course it's a bit simplistic, but the Kupffer cells seem to be very much equipped to participate in metabolism and to release inflammatory cytokines for innate immune cell activation. The monocyte-derived macrophages actually express a lot of markers that would be considered pro-fibrogenic or participating in stellate cell activation, but also in angiogenesis. And we are now at the moment trying to further develop this. So if you do an unbiased T-SNE analysis, you can identify several clusters of monocyte-derived macrophages. And what we're doing at the moment is that we do a single-cell RNA sequencing, meaning that you isolate the macrophage population of a liver. We started with mouse liver and very defined models. And then you actually the machine puts a cell with a single library that is barcoded, so you do a whole RNA sequencing of the transcriptome of every cell. And this is the first time I show our initial data because we don't understand them yet, but I just want to, I think this is really bit, we need to probably work on that if we really fully apprehend the heterogeneity of the cells and want to remove all biases that we have if we analyze our data, because we have a mindset, we think, okay, this is important for this and this is for that. Here we sorted cells from the liver that were negative for different lineage markers, also CD19, CD4, CD3, and non-neutrophils had a pretty good recovery rate. And this is what an unbiased analysis of these, I think, 20,000 transcriptomes look like. So cells seem to cluster. So there are different populations of cells, actually about 16, it looks from the first data. And if we overlay a chow diet and Western diet, we do see that the Western diet cells cluster around this area. So we have some populations that seem to be there only in a chow diet and some in the Western diet. We are not there yet, but if you look at CD68 expression, this is very prominent in this cluster. CCR2 is prominent in this cluster. So we do seem to have, or at least we think we will find our inflammatory macrophages or disease-driving or disease-characteristic macrophages in the single-cell analysis. But of course, you need the help of the bioinformatics, and I think here in Barcelona, you're extremely well equipped to do such analysis. Our bioinformatics facility is also now heavily involved in making sense of the data. Maybe from a more clinical perspective, what we already understand is that the recruitment of the monocyte-derived cells to the liver is actually an important step, not only for maybe translating into hepatocyte injury or mediating adipose tissue inflammation, but also for the process of activating hepatic stellate cells. There's a lot of indications that this is driven by the infiltrating cells. And of course, one idea would then be if you block the infiltration of new macrophages to the liver, but leave the Kupffer cells unaffected, that you might actually reduce fibrosis. And one clinically, or one drug that is in clinical testing at the moment is an orally available CCR2, CCR5 chemokine receptor antagonist. It's called CVC or Cenicriviroc. And if we use this drug in mice, so we were treating for 8 weeks an MCD diet, we're then treating for the past 4 weeks with the CVC or with the vehicle control, we actually see a very nice reduction of this red cell population, the monocyte-derived macrophages in the liver, while the Kupffer cell numbers are kind of less affected. And what was very striking is that we could not really see many differences with respect to steatosis or steatohepatitis, but a clear effect on fibrosis progression in this model. This is an MCD diet model where you see less of these red fibers in the group of mice that was treated with the chemokine receptor antagonist. We also tested a different model of Western diet. The disadvantage is that the Western diet model leads to a really steatosis and steatohepatitis, but not a clear fibrosis phenotype. So it's very difficult to learn something about fibrosis. But nonetheless, there was good inhibition of the monocyte-derived macrophages with the treatment of the drug, a good effect on the steatohepatitis phenotype, and also improvement of insulin resistance. So this probably tells us that not only the liver macrophages are affected, but also in adipose tissue and maybe also in the vasculature. So from this mouse data that less macrophages in the liver, less fibrosis, it's of course very tempting to go into the clinics, and this is a phase 2 trial that is ongoing with almost 300 patients. The design of the trial was that patients were randomized to either the drug or placebo, and then after year 1 they received the first biopsy, and after year 2 or after the year 1 the placebo group was randomized to CVC and placebo to give more to higher increase the chances of receiving the real drug in the trial. And what we have at the moment is the first year of analysis, almost 130 patients with sufficient biopsy data, baseline at year 1. I think a good mixture of fibrosis phenotypes, all obese, and the data look as follows, and it's very similar to the mouse data. So not really an effect on the histology of steatohepatitis, but a significantly higher rate of patients that improved their fibrosis stage with the drug. And if we look at the different stages of fibrosis, it actually seems to be a quite consistent effect. And hopefully for AASLD this year, the 2-year data, the final data of the study will be released. So we had, I think, the last patient biopsied sometime in July, so it takes some time to read out. I don't know the data yet, but I of course hope that this could be a new approach. Nonetheless, there is also another potential if you want to translate into therapies, because we know that the cells that are very pro-fibrogenic, pro-inflammatory can actually undergo a phenotypic change. So the macrophage can change the phenotype towards an anti-inflammatory restorative macrophage that would promote stellate cell apoptosis and deactivate the stellate cell. And of course, Pau has done some good work on understanding mechanisms of stellate cell activation, deactivation, and the different mediators included. And it's tempting to speculate that if you might switch the phenotype of the cells, that this could be very, very helpful. If you think about how to do that, one thing that makes it very easy if you work with macrophages is that you can target them relatively easily. So if you work on a new cancer drug, I think the majority of them fail because they never reach the tumor, because all the macrophages eat up what is floating around if the drug is just large enough. If we do that in mice systematically and use different drug carriers that have been proposed by the nanomedicine world, for instance, liposomes, microbubbles, or small polymers, and we see where they end up, at least in the beginning, a lot of them end up in the liver. And for instance, if we look here with liposomes, do the 2 photon, this time the Kupffer cells are in blue because we had only the liposomes in green, so we needed to change the color of the beads. Oops, sorry. You can actually see if you inject the liposomes in green, they really rapidly light up in the blue cells. So a lot of them is just like immediately after the first passage captured in the macrophages, which is of course their role. They need to capture bacteria, large particles. So it is possible, probably not to specifically target with unmodified particles, specifically Kupffer cells or monocyte-derived macrophages. If you look here at the liposomes, actually both are really targeted by those particles. But if we, for instance, put dexamethasone, a steroid, into the liposomes, we can sort of, at least in a first proof of concept study, reduce the number of fibrosis and deactivate the monocytes. And I think this holds quite good potential. Of course, you need to carefully think about what you want to modulate in the macrophage. And for this, you have to consider that the macrophage also changes the cellular metabolism upon activation. So, for instance, compared to a macrophage in homeostasis, a macrophage that is activated usually switches to aerobic glycolysis, produces ROS and nitric oxide, activates NF-kappa B, has inflammasome formation, cytokine production. And a lot of the drugs that we're currently discussing in the field of NASH will actually impact also on this cellular metabolism. So there might be some good ideas if you can target the macrophages and influence the pathway that you think is most responsible for this inflammatory switch, that this could be helpful in conditions of chronic liver disease and fibrosis. For instance, PPR delta is certainly upregulated in Kupffer cells, or the ASK1 inhibitor by Gilead is certainly also a very broad caspase inhibitor that would affect the macrophage metabolism. And in the end, of course, hepatocellular carcinoma is also affected by macrophages. And maybe just like from a conceptual point of view, you again have different functions of macrophages in the context of tumor. We understand very, very well that if you don't have macrophages, you actually have more tumors because probably very early tumor formation can be sensed by macrophages, especially cellular senescence, which then activates T cell responses. So for recognition of early tumors, macrophages are very important. On the other hand, we know that the macrophages that we see in an HCC patient are probably pretty bad because they are usually suppress immune responses, provide a rather tumor-friendly environment with lots of blood vessels, and express IL-10. So these are probably cells that we don't want in the tumor environment, especially not if we think about new immune therapies where you want to block tumor cell T cell interaction, but maybe also suppressive effects from the macrophages to enhance immune checkpoint inhibition. If we look in a fibrosis model without a liver tumor, and we do a micro-CT analysis of the blood vessels, this is a reconstruction, 3D reconstruction. You can see that in a fibrotic mouse, you have a large increase of blood vessels. This is one of the hallmark in renal fibrosis. You have less blood vessels, but in liver fibrosis, you have an increase of blood vessels, which probably is a key point for a tumor-friendly environment. When we isolate the different components or cellular components of a fibrotic liver, some of the angiogenic factors are really highly expressed in the monocyte-derived macrophages. So one idea would, of course, also be what is the effect if you inhibit those monocyte-derived macrophages in the context of fibrosis or maybe tumor development. What would it do, for instance, to angiogenesis? And this is shown here. MNOX is an inhibitor of CCL2. It's a little bit easier to apply than the chemokine receptor antagonist because you can inject this subcutaneously three times a week. We can actually almost normalize the blood vessel formation, at least by my micro-CT scan or by CD31 staining. We are unfortunately not equipped for hemodynamic measurements, so this is something that we never did, but we always wanted to understand whether there is also then an impact of the monocyte-derived macrophages on the hemodynamics, and maybe this can be addressed together with all the expertise in this room. Nonetheless, if you use it in a combined fibrosis tumor model, the model that we prefer is to inject DEN very early in life at 14 days of age and then inject CCL4 for 16 weeks because this gives you multiple tumors in an injured liver. So it's not perfect, but it's at least better than having a tumor in a completely normal liver or having something genetic knockout that never appears in human. And if we treat here with the with the CCL2 inhibitor, we actually do see at least much less vascularization, and it seems to correlate with tumor volume. So this is at least our preliminary data at this point. So with this, I think I gave some ideas for discussion, and I am very grateful for the many input, discussions, and interactions that I had today. It was really great, also with Alicante and Barcelona team. Just to summarize what I think one should know about macrophages in the liver, we have different types of macrophages. The Kupffer cells that are there in the sinusoids sensing injury, and this can be LPS, this can be DAMs, danger-associated molecular patterns from injured liver or from injured hepatocytes, and that will signal to stellate cells, but also immediately release a lot of cytokines that injure the liver, and CCL2 among many others that recruit immune cells to the liver. But then the liver monocyte-derived macrophages are actually the dominant population of macrophages in the liver, and they do a lot of bad things like angiogenesis and activating hepatic stellate cells. There's also the possibility that this is even amplified by proliferation. There's some good indication that both populations can proliferate. Nonetheless, if you stop the injury or if you provide the right stimuli, those cells can undergo a phenotypic change, deactivate, and then also deactivate stellate cells, probably also block further cell injury. I would like to thank the people from my group who did all the work that I showed you here. A very active group. I'm very happy. A lot of collaborators. With some of the collaborations just started with Jordi. I hope we can really expand this. And I would like to summarize. The macrophages are a key population in driving inflammation, fibrosis, and probably also tumor microenvironment in the liver. And the interesting but also the complex thing is that the cells are heterogeneous. They respond to different stimuli, can act pro or anti certain or disease progression and promotion. There are some good hints that we can therapeutically target. I showed you the example of blocking the infiltration of monocytes to the liver, but potentially also switching the phenotype of the macrophages that are in the liver. And some areas of research is how to balance this, maybe also how to image the interactions of cells that is going on. Of course, what is really happening in cancer, what makes a pro or anti-tumoral macrophages in the setting of cancer, new nanoparticles, and of course the translation into the clinics. Thank you very much. Yeah, and also the penetration is of course different, because it's probably very important to block the cells that are infiltrating in the periportal area, but if you have already very established cirrhosis, this might be difficult. We have never tested this because the mouse models are really not convincing for cirrhosis. It's you don't really. I mean, we have a hard time establishing portal hypertension or so in mice. The rat models would be much better to do that. It's a bit open. Nonetheless, I think the effects will probably be less pronounced than if you do it in an ongoing, let's say, maybe in human F2, F3 transition. It's probably much easier to be effective than in F4. Yeah. Well, the thing is, it's actually not so easy to answer this with the mouse data that I have, because the affinity of this antagonist to CCR2 and 5 is about equal in humans, but for mice it's a very, very strong effect on CCR2 and a less effect on CCR5. And so I guess what we do see consistently is that, for instance, if we do chemotaxis with lymphocytes, and lymphocytes would rely on CCR5 interactions, we block with this receptor. If we look into liver injury models, and we've looked at several models, we do not see any effect on lymphocyte migration in vivo. But we do see a clear, very, very specific effect on the macrophage migration. We even, if we do an unbiased TSNA analysis, which you put all cells in that you detect by fax, and you then ask, okay, what's different in my treated group versus the untreated? Only the monocytes come out as the population that is affected. I think in the mouse model, it's a very specific effect. In humans, or it might be much more pleiotropic, I fully agree with you, than what we model in the mouse model. If we treat stellate cells with which would also respond to CCR5, we cannot really influence collagen secretion. But this might be also to the fact that not everything what will happen in humans can be reproduced in the mouse model. In human cirrhosis, first of all, you have, even in advanced cirrhosis, if you look at explants, you have a really very, very high number of macrophages. So it's not that you could only say, well, it burns out or it decreases after a while. So this seems to be pretty consistent. If you look at the markers, it's unfortunately not so easy to determine the origin of the cell in human liver. What is apparent is that CD16, which is a marker of non-classical monocytes, which is an FC gamma receptor, is highly upregulated. So arguing for really sort of a monocyte-derived population of the human macrophage in cirrhosis. And CD14 is also present, which is an LPS receptor, not not so much expressed on Kupffer cells, but more on infiltrating macrophages. So in human cirrhosis, there seems to be a higher number of infiltrated macrophages rather than just expansion of tissue-resident macrophages, and they do seem to have an inflammation-driven phenotype. But the idea is, of course, to use the single-cell RNA sequencing then also for human explants and healthy human liver, if we can get from resection of colorectal metastasis, and then really assign the different populations that we find in mice to the human counterparts, because that is not so clear at the moment. Yeah, and I think this dichotomy of Kupffer cells and monocyte-derived macrophages explain this probably, because the Kupffer cells, they actually are the ones that eat Staph aureus. If you inject it into mouse fluorescent Staph aureus, they are captured by the Kupffer cells. But the monocytes, they have less scavenger receptors that are not, and they are probably not ideally located at the luminous side of the endothelium to catch the bacteria that are floating around. And the fact that with the CCR2/5 inhibitor, we do not see any increase in infectious complication. That was a concern, that you might be more prone to sepsis or infections overall, is probably that you don't affect this resident population of cells that are still able of scavenging what is around there, but the inflammatory component that is freshly recruited and less prone to phagocytosis, this population would be monocyte-derived. Yeah, I would expect that there is a lot of plasticity between certain populations within this big macrophage pool that all express CD68. What we have done in parallel, but we have not run the Illumina kits yet, is to study also bone marrow-derived or bone marrow cells from a Western diet and healthy chow diet fed animals, to understand the heterogeneity, if we can relate certain populations also to changes in the bone marrow. But I think it will require much more analysis at different time points to see how this really progresses over time. And also we need to study different models in parallel and see what is really consistently there and what is maybe really much driven by one model or the other. Well, it is known that this can happen if you deplete Kupffer cells in mouse models completely. The monocytes can come in, and after a while they acquire a phenotype that you cannot distinguish anymore from the Kupffer cells. And probably this is also true for functions like phagocytosis. What is not so well known at the moment is whether this really happens in injured liver or not. And it probably depends on signals from the endothelial cells, for instance, that they provide a certain signal that there's a niche, there needs to be a Kupffer cell now at this position. But in an injured liver, we know the number of Kupffer cells goes down, at least their relative numbers go down, and the monocyte-derived cells prevail. But how much of the monocytes can actually become benign Kupffer cell is not so clear at the moment, because also the Kupffer cells are activated in NASH. It's not that it would be a disease that's only driven by the cells from the outside, but the cells that are inside already are also activated, respond to the injury and change their phenotype. But in principle, this can happen, and this, of course, gives hope to the assumption that you could maybe use a cell therapy. You could maybe isolate Hymis macrophages, culture them, keep them a bit in a healthy state, and then inject them into a patient, or maybe even autologous cells from a patient. The researchers in the UK, Stuart Forbes, will be here. I think he will present you some data. I'm highly skeptical because I think it's very, very dangerous, because we know that the macrophages are so plastic. I think it's very, very, very difficult to keep them in this anti-inflammatory state. It's much more likely that they will adapt the phenotype again, and if it's needed to be aggressive, they will be aggressive in the environment. It would be much better to use the endogenous macrophages that are there and try to repolarize them. Thank you very much. We have not done mouse work with alcoholic models, and unfortunately we don't see the patients, or maybe fortunately we don't see the severe alcoholic hepatitis that often in Germany. We see more decompensated alcoholic cirrhosis. Nonetheless, there are animal work that show that basically alcohol is also toxic to the macrophages, but the sequence of event that was discovered by Cynthia Ju from Denver, she now moved, I think, to Houston, is very, very similar. So I would interpret that the negative effects with the anti-TNF was really based on the deactivation of the Kupffer cells, that this would really be a setting where the patients are very prone to bacterial infections, and if you target then very broadly the macrophage functionality, that this is too broad and too dangerous. Probably if you would use an chemokine receptor antagonist, this has not been done to my knowledge, it might be a bit safer, but probably also a bit less effective in such a really overwhelming disease condition as alcoholic hepatitis. But I think the Kupffer cells, this is what I take out from the infliximab trial, was kind of, if you block too harshly, too broadly with less selection in the patients, you are at risk. Thank you very much for this question. Unfortunately, I think that the monocyte-derived macrophages do not represent the majority of peritumoral tissue macrophages in HCC, at least from our point of view. We have, for instance, studied this model of DN plus CCL4 with the chemokine receptor inhibitor, not the receptor, the chemokine inhibitor, and we see a reduction of the peritumoral macrophages by about 20%. And if we stratify the different populations, there has been one paper from Joe van Ginderachter's group in Brussels that showed different populations of peritumoral macrophages. And it seems to be that this inflammatory component is only a smaller proportion of the peritumoral or tumor-associated macrophages in HCC. So the effect in a fibrotic liver is much stronger than in a tumor-bearing liver. I thank you again for being here, for being the first speaker of the liver seminars. Thank you, Madrid. See you next month. And thank you all for being here.
Great, thank you.